mostly and stuff.
All right. Good morning, everyone. Welcome back to our seminar series on Translational Biomedical Engineering. It's a great pleasure for us today to host Marc Thurner, the CEO of mimiX Biotherapeutics. So Marc has joined us from Switzerland. So it's evening, and then Switzerland. Thank you very much, Marc, for accepting our invitation. But before we start, and as usual, if you have any questions during the talk, please make sure that you use the question box. Ask a question box is right in the bottom panel of the screen, and you will be able to share your thoughts and questions with us and then also our speaker. So our next week speaker will be Professor Farshid Alambegi, who is an assistant professor at the Department of Mechanical Engineering at the University of Texas, Austin. And then he will be talking about intelligent and flexible robotic systems next week. If you want to get most up-to-date information about these e-seminar series, please make sure that you follow us on Twitter. Our handle is TransLBME. You can always contact me and Human in case you wanted to get more information about these series, if you want to nominate the speakers, and if you have any comments or feedback about these series. Last but not least, we would like to thank Montreal TransMedTech Institute, who has been with us since almost the beginning of these series. They have been supporting these e-seminar series so far with 75 speakers. So thank you, Montreal TransMedTech. So with that, I'd like to introduce our speaker today, Marc Thurner, who is a visionary entrepreneur and pioneer in biofabrication technologies. He's currently acting as the Chief Executive Officer at mimiX Biotherapeutics. He pioneered the field of 3D bioprinting with his former creation of regenHU, which is today recognized as an industry leader in the development of innovative biomedical solutions. A microtechnology engineer graduate, Marc started his professional career as an R&D engineer, managing international projects for high-precision automation industry. His avant-garde view that the future of regenerative medicine lay in converging engineering and biomedical science brought him to found regenHU in 2007 and mimiX Biotherapeutics in 2020. In 2012, Marc created the regenHU spin-off Vivos Dental, specialized in innovative 3D printed bone grafting solutions for oral bone augmentation. Being an industry transformation leader, mimiX Biotherapeutics develops leading-edge solutions in biotechnology and tissue engineering that enable scientific and clinical breakthroughs. This Swiss-based company collaborates with a global network of scientific innovators and industry players and leads the development of biomedical products for regenerative medicine and drug discovery. Marc Thurner and the mimiX Biotherapeutics team are passionate about addressing the current and next generation biotechnology challenges that help the progress of life sciences. So the company and Marc, all of them, they have made tremendous achievements and recognitions, including Venture Leader Award, IMD, EMBA Startup Competition Award, and Industry 4 Sharpers Awards in the different years. With that, I'd like to thank Marc for accepting our invitation again, and then I would like to give the virtual floor to you now, Marc. The floor is yours.
So, hi everyone. Thanks, Moshe. Thanks, Human, for the nice introduction. Thanks for giving me the opportunity to present mimiX Bio and its sound-induced morphogenesis technology. We are a young company founded in 2019 with our headquarters in Switzerland, and we aim to make regenerative medicine accessible to everyone. And we address this challenge with a novel biofabrication technique named sound-induced morphogenesis that has the potential to overcome some of the major limitations in regenerative medicine. mimiX has two business units, a scientific instrumentation business unit that brings this platform technology to the scientific and R&D community, and a clinical translation business unit that focuses on four focus areas, namely wound healing, cardiovascular, oncology, and trauma and orthopedics. The technology we built on was invented and incubated at the EU Foundation, a medical guided organization that promotes excellence in surgical treatments in trauma and musculoskeletal disorder. Organization that has very likely one of the most extensive network of physicians with plus 200,000 members, and we take advantage of this community to translate our technology into clinics. Our technology, sound-induced morphogenesis, is a convergence between art and science. It's a novel biofabrication technique that builds on two very simple physical laws. The first physical law is the law of mechanical vibration that we use to move particles. And we create these mechanical vibrations with sound waves, with energy generated by sound. And by controlling the profile of the sound waves, we can not just move these particles, we can pattern them. The second physical law is the law of static waves that allows us to make this particle patterning in liquids and hydrogels. And we converge these two concepts in one single process unit that we named cymatics, and that enables, in a very easy, ultra-fast and scalable manner, the creation of two-dimensional patterns of cells, of organoids, of microtissues, spheroids, but also non-biological particles, any type of particle. And we cross-link the layer and then go 3D by building the next layers. So we master pattern formation, and patterns are at the start of everything in nature. Even life, we are the result of embryogenesis, morphogenesis, that starts with cells that condense along a specific pattern. So it's all about shape and function. So if I summarize the step that you did see on this movie, on the left you see this tabletop device in which we place a labware. This labware is loaded with the biological cocktail that comprise cells and hydrogels or cell culture media. And by applying sound energy, we condense this particle. So we create an agglomeration along a specific pattern. And once we have the pattern of interest, we cross-link either through a thermal shift, through a photo initiation, or through the addition of a chemical component. So we coagulate that layer and go to the next layer and build a 3D object layer-wise. So sound-induced morphogenesis is a newcomer in the biofabrication toolbox. And it extends this toolbox that already comprise quite a large spectrum of technologies. Here, I just want to show you how sound-induced morphogenesis differentiates from the other technologies. As introduced, we build a layer object in two steps. First, we condense the material and create the pattern. So the starting point of sound-induced morphogenesis are cells that are fully or randomizedly distributed in a hydrogel. And through this energy, we create a local cell density enhancement where the cells are forced to come together and to talk to each other. So we initiate or facilitate the cell-cell interaction. If we compare this approach to the bioprinting that starts with the same initial situation where cells are randomly distributed in an extracellular matrix hydrogel, we press this solution through a needle or nozzle so that at the end we have a strand that, if we have a closer look to the repartition of the cells, are similar than in the initial situation. They are randomly distributed in this ECM and are not forced or invited to talk to each other. This talk to each other cell communication is emphasized in the organoid and spheroid manufacturing approach. If we have a closer look to the hanging drop approach, as example, where the cells are delivered in a droplet in which, through gravitational forces, the cells are invited to talk to each other and to create agglomeration and form a microtissue. So that's exactly what we do on the sim level. And if we go into the world of organoids, we can even control different or multiple cell types that in the standard approach follow a randomized morphology or development process. While if we apply this organoid concept to the sound-induced morphogenesis approach, we create a first circle, for example, of condensed cells, and then with a second cell type, we create a second condensation around the first one, and we can create control the morphology of larger scale organoid formation. So we have a newcomer extending the biofabrication toolbox that mirrors nature's design strategy by controlling the shape and the function. Let's have a look on how this looks real time. So on the left, you see a short movie showing the sound-induced morphogenesis technique, and on the right, the conventional 3D printing approach. And the goal here is to create a honeycomb structure out of a calcium phosphate formulation. And you can see on the left how easily, just by triggering two parameters, frequency and amplitude, we can create the pattern. It's fully contactless. It's contactless, it's just through the sound energy. And I have also to say that it's sound in a low frequency range. So we use frequencies from 0 to around 600 Hertz. So completely compatible, cyto-friendly. And we can, in an ultra-fast and scalable manner, create this architecture. And once we have the architecture of interest, as mentioned earlier, we can cross-link, photo-cross-link. We cover the entire spectrum of cytocompatible photo-cross-linker from 365 nanometer up to 520, or through a thermal shift or through a chemical addition. So if we have a look at how this biofabrication industry has evolved over the last 30 years, an entire workflow that has the mission or has the ambition to move from a 2D environment to a 3D environment that is more in vivo relevant. And along that workflow, multiple technologies like liquid handling, where liquids and particles are manipulated, bioprinting that brings the shape fidelity into the game, mini organs, spheroids, organoids that on a small level are in vivo relevant, and microfluidics that brings the dynamic dimension into the technology. But it's nowadays recognized that very likely none of these individual technologies has the potential to overcome by its own the limitation of tissue engineering and regenerative medicine, mainly given to the fact that very large cell quantities are needed, that the in vivo relevance is limited, that the capacity to induce micro and macrovascularization is limited. And that's where we see the big step that sound-induced morphogenesis introduces, that in its simple process has the DNA of each of these individual technologies. We handle liquids and particles. We go 3D, like bioprinting. We enable this organoid and spheroid in vivo relevance through the process that I showed earlier. And we can integrate microfluidics in the labwares to enable perfusion, to enable dynamic fluid exchange. So we integrate each of this unique technical feature of these individual techniques in one single process unit. And this opens the door to a large field of opportunities, say it in life science. But there are other fields that are very hot topic related to large cell and large scale cellular constructs. The example of clean meat and the entire industry around cellular agriculture. So let me show you a few real examples. A first very simple example, forming circles of vascular cells. So what you see here, these are UVEC, vascular cells that are condensed along a circular pattern. And post-processing, after 5 days, we can see how this preformed pattern self-assemble in and forms a highly dense microvascular network layer. If we have a look of how this is manufactured, so this is a real-time movie, you can see that within a few seconds, these circles appear. So you have here an inner circle and outer circle that is formed within around 35 seconds. And as introduced earlier, once you have the pattern of interest, you cross-link and can go to the next layer and or mix into this first layer a second cell type or a second type of molecule that you can pattern in a B model mode. So you can see now after about 30 seconds, not even 30 seconds, we clearly see these two individual formed cell agglomerated circles. So why is this cell agglomeration so important? Let me show you an example using cardiomyocytes. In the upper example, cardiomyocytes are randomly distributed in a hydrogel, while in the under example, the cardiomyocytes are agglomerated along a circular shape. If we have a look at the energy mapping on how these cardiomyocytes react, you can see that they beat in a completely unsynchronous manner. Each one is doing his own job. While when they are condensed and form a pattern, and what you can see here is a stronger gray line, which is a cutout, a magnification of a part of this circle here. You can see when we do the energy mapping that they communicate. They deliver a signal in a synchronous way. They beat together. And this is only possible through this local cell density enhancement and delivering this signal along the pattern of interest. Another very important aspect of the technology is the capacity to create large-scale functional macro and microvascularization. And what you see here is a pattern formed out of UVEC cells, where during the manufacturing process, we created a macro pipe that is later used for perfusion. This is a circle of around 4 centimeters, and you can see that 3 or 4 days post maturation, when we perfuse a media, in that case dextran, these micro capillaries are fully functional up to the border of this 4 centimeter circle, where you can see how this dextran is delivered through this entire surface. So it's a proof of concept that this approach enables the creation of functional macro and micro, highly dense, I have to say it, highly dense networks. The other point, or the other magic point, is when we condense the cells, when we play on this local cell density enhancement, we can significantly reduce the quantity of cells needed to do later the biological job. And we can, through the condensation, reduce the cell quantity needed. And on the other hand, we can go larger scale, having a more physiological relevant system. So less cells, bigger constructs. And this is a major topic in cell therapy, knowing that nowadays the most common cell therapy route starts with a biopsy that you process in a GMP lab, where the cells of interest are isolated. You get a few thousand, but you need billions. So you have to amplify and amplify for weeks. At each amplification step, you affect the quality. So if you can reduce the quantity of cells needed for the regenerative step, you are in a pole position to facilitate the logistic aspect, the quality aspect, and the delivery time of the therapy. Sound-induced morphogenesis is not only interesting to pattern particles, it can also be used to pattern extracellular matrix components, to densify, to pattern, to orientate fibers, to create, for example, gradients, mechanical gradients, or mechanical niches to support cellular development. So I could show you plenty of other examples. My ambition today is not to give you a scientific talk. I would not be the right guy for this. I would invite our CSO. I just want to show you the opportunistic arena that this novel technology offers. On one hand, it speaks the industrial language. It's super easy, contactless, so fully cytocompatible. It's scalable. We can go large scale. And it speaks the industry and the clinical language. And on the other hand, it offers a few assets to overcome the current limitations of regenerative medicine. It enables the reduction of cell quantities. It enables the manufacturing of highly dense micro and macro vascularization networks. And it enables multi-layer stacking. So not just to have the cell-cell communication on a layer level, but cell communication in 3D. So we offer or make this technology accessible to the scientific and R&D community through a platform product named Cymatics. So it's a tabletop device that looks, in terms of dimension, like a coffee machine that you can either use as a tabletop device or place in a biosafety cabinet. This device has different cross-linking strategy. We have UV lamps to cover the entire visible or cytocompatible spectrum from 065 to 520 nanometer. We have a thermal controlled plate to use thermosensitive materials, and we support this platform product with a world of labwares and a portfolio of biomaterials. Has to be said that most of the well-known bioprinting or bioink portfolio that we know for decades now from the bioprinting industry can be used with sound-induced morphogenesis using dilution protocols. So it's not starting a fully new world of biomaterials. It's exploring the already well-known. So to finalize my presentation, I just want to give you a short insight into our bioconvergence agenda. So we are working on the convergence of the SIM technology that offers the best biofidelity. So if I go back to the statement of mirror nature's design strategy that offers optimal biofidelity with the shape fidelity. And this convergence is the result of the convergence of sound-induced morphogenesis with additive manufacturing. And we work on and use in our internal R&D activities probably the most advanced biofabrication solution, which is the result of this technology convergence. And when I say it's probably the most advanced, it's a solution that brings the entire biofabrication toolbox in one solution, say it sound-induced morphogenesis, bioprinting, organoid manufacturing, microfluidic, and this in a lab automation setup. So let me show you how this bioconverged approach can be exploited. I show you an example out of the regenerative medicine scope. Again, back to what I introduced earlier as a high topic, clean meat. So with this novel platform, we can manufacture in a parallelized way layers, cell layers, in that case, muscle layers, layers of muscle cells, of fat cells that we can enhance with 3D printing. And this in a lab automation format where we can then individually handle these layers for future maturation step and for stacking. So this is just a teaser to show you the level of opportunity that this technology converged in one single device opens. So I'm at the end of this presentation. I thank you for your attention. I invite you to have a look at our website, mimixbio.com. We present use cases, we present SOPs, so have a look at it if you're interested to learn more. And I also invite you to follow us on our social networks. I have also to say that we are constantly looking for strategic partners, say it's in R&D, early stage R&D, in clinical translation, or on industrial translation. Thank you very much for your attention.
Thank you so much, Marc, for this interesting presentation. You can stop sharing. So, maybe the audience can see the information on the screen and follow your company on Instagram, Twitter, and in LinkedIn for a couple of. Okay. So, and then before going to Q&A, I just want to go through our next speaker, so you can have some time to take a breath and refresh yourself. So next week we have Professor Farshid Alambegi from the University of Texas at Austin, from the Department of Mechanical Engineering. He's going to talk about self-engineering using intelligent and flexible robotic systems. So if you want to get the updated information about our e-seminar series, you can follow us on Twitter with the handle TransLBME. So right away, I go to the Q&A box. We got several questions. The audience made my life easier. So first, I start with the question of Professor Hosli. Bonjour Marc, ça fait longtemps qu'on n'en a pas discuté. Can you comment on rheological limitation of the acoustic approach? Can the cells be placed in any type of fluid? Perhaps I missed this. I joined 9 minutes.
So hi Corinne, it's a pleasure to read you. Yeah, it's a long time, definitely. So as said during my talk, as long as we can create the static waves that are generated by the sound generator, we can pattern. And the rheological limitations are given through the viscosity of the material and the particle dimension. So there is not a number that I can claim today. It's as long as you have the possibility to create these static waves, you can pattern. Yeah. So it opens the door to most of the medias, of cell culture media. So you can create this organoid formation showed in the movie. In cell culture media, we use fibrin, we use collagen, we use alginate, we have synthetic hydrogel. By the way, the materials that we offer through our proprietary portfolio are synthetic hydrogels in the form of kits where you can fine-tune the chemistry, you can add some degradation sites. I invite you to have a look at our website.
Thank you so much, Professor Hosley, and thank you, Mark, for answering the question. So the other question from my co-organizer, Professor Akbari Mohsen. Can this technology be used for volumetric bioprinting? Also, what are your thoughts on using sound-induced morphogenesis for multi-material bioprinting?
So the answer is probably yes. I have to say that today we load just with a pipette the media into this labware. So you could use any type of dispensing device to make that job. Volumetric sounds to me 3D printing, and this is in line with the last slides of my talk, the bioconvergence of sound-induced morphogenesis with additive manufacturing, and that's definitely opening a fully new world of opportunities because there are some materials that you can definitely not process with sound-induced morphogenesis, and you can start to explore the best from biofidelity through sound-induced morphogenesis and create 3D structures with additive manufacturing.
Perfect. Thank you so much. So there is a question from one of our audience, Vahid. Maybe this is not a right question to ask, but he's asking you if there is any limitations of your technology in terms of 3D in terms of 3D printing compared to the other platforms or other technologies. So, it's a very difficult question to ask somebody who is selling a technology.
It's not a difficult because any technology has its limitation. And this is also the case for sound-induced morphogenesis. We have a beautiful technology to do this shape and function control, agglomeration and patterning, but we cannot claim that we can do 3D constructs in a free form like 3D printing. So yes, SIM has its limitation, but we are overcoming this limitation through the bioconvergence. And that's why in this super advanced cutting edge device that I showed on my last slide, we converge sound-induced morphogenesis with additive manufacturing. And that's a game changer.
Perfect. Perfect. There is another question. I think you already answered this in your presentation, but we can review it. Mahsa is asking, have you ever made different types of layers of biopolymers as ECM?
Yes. Yeah. So today we can go up to around 7 layers. Then it's depending on the size of the construct and the thickness of the construct. But we can do multiple layers up to around 8 to 9 millimeter heights. Yeah.
Perfect. That's interesting. So again, another question from Masa, maybe follow-up question. Why cell quantity or density is important? Is it important? Because you said that we need less cell.
Yeah, so it's important. I said, so the cell therapy world says less cell we need, less we have to amplify, faster we are ready to deliver a therapy. So at the end of the game, it's about reducing the time from the cell extraction in the biopsy and delivery of the therapy. And this is usually X weeks. If we can shorten it by reducing the cell quantity, you have a winner. And that's what I say. Sound-induced morphogenesis has the potential to rethink some of the major problems in the current cell therapy process which is the long delivery time, because you have to gain these cells through the amplification.
Perfect. Thank you so much. So Helen Bernie is asking, is there a way of designing the pattern that you create? Is it always circles, or you can have different
No, it can have different shapes. It can have different shapes. You're right, I showed only circles. Sorry, I will improve that in my next talk. But you can not do everything, but you have quite a large spectrum of patterns that you can form. And at the end of the day, it's not to have the best pattern. It's about offering the best starting point to the biology to do the job in post-manufacturing, in the maturation. So it's about creating these organoid models, condensation and patterns in an efficient way.
Perfect. So these are more scientific, technical questions, but now, because you are kind of professional entrepreneur, you already created 2 companies. One of them was very, very successful, regenHU, the first one, and you have been in the business for more than 15 years. So now MS, that's probably a acronym. Hi, Mark. What is the regulatory pathway for the device, and how far are you down?
I love this question. I love this question because
yes,
the 15 years of experience I share in this industry, it's all about being able to translate the beautiful things that are done in science. You can do space shuttles, but if they cannot be translated in a humanistic way into clinics, it will limit his lifespan to a nice publication. And that's where one of the beauty of sound-induced morphogenesis is in the simplicity of the process. So if you consider the regulatory pathway, and what I did not say in my presentation is what we are doing in our clinical translation business unit. So I hope to be invited in 6 months from now and to be able to present you the things that we are doing. For example, we are working on a new skin graft that is produced point of care. So point of care, in a regulatory point of view, we take only materials from the patient. We do not go out of the surgery room. And our printer is a medical device class 1 here in Europe. So it's more or less the same in US. It's a medical device class 1. And the regulatory complexity is placed in the labware, which in the case of that skin graft is a medical device class 2B. And from a regulatory pathway, it's very, very straightforward. It doesn't fall under the rules of ATMP. And therefore, we see a quite sexy clinical translation pathway. I think you are mute. I can't hear you anymore.
Yes, because yes. So I'm sorry. On the same line of the business aspect, Maso is asking, what exactly is your value proposition?
So the value proposition of mimiX is a game-changing biofabrication process. And if we look at the economical pathway, so we have these 2 business units. One is about commercialization of the platform in academia, but we all know that academia is not the market. So it's not the value proposition that mimiX intends. We want to explore with academia the potential of the technology in as much we can indication fields that we later translate to clinical or therapeutic use through our clinical translation business unit.
Perfect. Thank you so much, Mark. And a follow-up question, I don't know if this is relevant, but how this technology will increase the value of cell manufacturing?
So how this will increase the value of cell manufacturing. So cell manufacturing is today facing the question of quality and scale up. And so if the technology that we offer can reduce the quantity of cells needed and you have less amplification steps, you have a better quality, the simplicity of the technology speaks the regulatory and quality language. It's so simple, it's reproducible. So I think all these technical features are speaking toward improving the cell manufacturing or the therapeutical manufacturing language.
Perfect. So now I want to ask, the questions, my own questions about your experience to become an entrepreneur. Because we have lots of students and graduates who wants or would like to become an entrepreneur, and they want to know about your experience as somebody who has more than 15 years of experience. So what made you kind of become an entrepreneur after your graduation? How did you start? And where did you start?
I worked a few years in the industry, in the biotech, in the machining industry, and I think it was in my genes since the beginning. I wanted to found my own venture. What I did not know, and maybe this was also the success of regenHU in the early days, I was naive in biofabrication. So I did not fully understand the complexity of the bio-material cell environment. And this allowed me maybe to go in directions where others knew how complex it will be and decided not to go there. So we did a few strategic moves with regenHU, and the same now with mimiX. That certainly builds also on some opportunistic mentality.
And have you ever failed?
Oh yes. Every day is a learning process, and every day.
What are the challenges of
the day, there is a beautiful concept that comes from the Silicon Valley that is named the minimal viable product. It refers to a physical product per se, but if you look at the steps behind it, it's build, measure, learn, build. It's a loop. So you go out with an idea, you build that idea. It can be a starting point, a PowerPoint file that you present. You see the feedback, and then you improve. And that's the process that I personally use in my daily life. You never have the perfect idea. The client, the community that is using device today, the cymatics, has the great ideas, and we try to improve the product's performance, the capacity of the device to enable all these crazy ideas to become reality.
Nice. Thank you for sharing your experience. Professor Corinne Hosli asking on the same line a question about how did you raise funds to start up these ventures? What was your initial cash flow? Was it more governmental or soft money or grants or non-dilutive, or when did it switch to the actual sales?
Yeah, so money is the oxygen. So money is for a company what the oxygen is for a human. So if you run out of oxygen, you die. So the same reality for a company. So to get enough cash to reach the next value step is usually a big challenge. There is a mix between public granting money and private money. The public granting money is for me a kind of very dangerous tool. It's very sexy, it's accessible, but very often it's subject to decision leaders that do not understand fully the industrial reality. Yeah. So getting too much public money, I don't want to say getting public money, too much public money is taking you in a direction that might be completely at the opposite of the reality. So my vision as entrepreneur, or what I do today with mimiX, is not to focus on public money. It's to work with the guys who can make an impact in industry and clinic. These are physicians, these are leading scientists, these are leading industry players, and joining efforts with this market leader or industry leaders gives also us access to financial possibilities.
Thank you so much. I have a question. Now, I mean, two ventures or two companies that you have, I just want to know what you will not do if you go back 15 years in the time, and you will not repeat it, or the mistakes that you did in making the companies or any move. Can you just comment on that?
Yeah, there are a lot of mistakes or a lot of learnings. I would call it learnings, because entrepreneur has to take decisions. Either you go left or you go right, and you take the decision based on the information you have the day you have to take the decision. So we are always more intelligent when we can look back. So that's why I say not mistakes, it's a learning process. There is probably one learning process that I did implement at mimiX. It's to build a strong governance, a strong advisory board, a board that comprises people who have this plus 20 years of industry experience, and that brings with them the network where they just have to take the phone and open the door to a venture capitalist or to the Harvard professor and so on. So finally, it's all about the team. And you need the right equilibrium in the team, not only scientific guys, not only engineers, not only business people. You need the optimal ecosystem.
Perfect. I'm asking this question because
I want to say greetings to a good old friend who I learned a lot from. It's Eric Gattenholm and the Selling founders. We were competitors for years. They showed me that they learned me the American mindset, and I was with my Swiss mindset. And my former company still evolves with the Swiss mindset. And I learned from them, and I hope that in a few years you will see mimiX as the #1 bioconvergence company.
So you are not competing, you are friends.
We are only friends, strategic partners, great value.
We had Eric in this seminar series. I think it was in the beginning of the last year. And he was talking about the bioconvergence and what they are doing with the cell ink. And I mean, both technology and both company, regenHU and cell ink, I worked with the regenHU machine when I was at University of Toronto myself. I don't know if you remember that you installed one machine also there. So anyway, yeah. So. And I mean, now in my lab, I have cell ink, like a small desktop machine, but hoping to have the morphogenesis also in the lab if we have, if we raise some funding. So, Marc, I'm asking this kind of questions about your experience in entrepreneurship because of the students, graduate students. So one last piece of advice for those who are going through entrepreneurship pathway from you, what would you tell them, to do or not to do, if they are taking this path?
Yeah, so I could make a talk only on that topic. Yeah, so to give a.
So I have to invite you two times, because you said that in 6 months you will give us more information, and then maybe one talk about the entrepreneurship. Yeah, it's our
one learning or one feeling that I see over the last years, that every PhD that ends its PhD wants to create its startup. Have a startup. It's like the most beautiful thing in life. But having a startup is a very, very, very hard time. A very hard time. You have to make huge sacrifice. Say it your first startup, say it the second one, even my mimiX now. It's a huge sacrifice in terms of time I spent with my family, with everything. So founding a startup is not the big dream per se. It's a hard work. It's a beautiful journey. And what I would suggest to all these PhDs or these students ending their training, it's work in industry. Go to industry and feel what industry means, what earning cash to feed the company, to feed your employees means. Yeah. And once you have the scientific background and the industrial experience, you're ready for the job.
Fail early,
right.
Fail cheap.
Yeah.
That's that's the term. So
in Switzerland, you know, in Switzerland we have great chocolate, we have great watches, and we have good tennis player. And Wawrinka has a tattoo written "Next time, fail faster," and that's what I learned from tennis. I don't play.
Exactly. No, that's true. So I wish, you know, there was no COVID and we had you here in Montreal. Are you planning to come again in Montreal? Or
yes, I hope to have plenty of partners in Montreal. You having a cymatics, Coring having cymatics, and I hope many others.
Yeah. Or maybe me and Coring can buy together.
It's the entrepreneur.
Okay, Marc, thank you so much. It was I really enjoyed your presentation, and I have been following the technology during the past years, and I'm excited to see that you're kind of, you know, bringing this to the market for more users. And hopefully, I wish you all the best, and I'm sure that we will see the same success that you had in regenHU during the last 15 years. And looking forward to hosting you here in Montreal with Professor Hosli and other colleagues, and Sophie Lerouge, my former supervisor, and so we can have 5 cassettes together one day. So, Marc, thank you so much. Hopefully, in next 6 months, I will contact you to get more story about your technology. Great. As you mentioned.
I promise. And I will be in Toronto live. Thank you very much. And
thank you so much. And have a great evening in
thank you.
Switzerland.
Have a good day.
Take care.
Bye-bye.
Bye-bye.
Everybody.
Bye-bye. Bye, everyone. Take care.